All right, so recording has just started. Now let me start the screen share and let's start with our thing. Okay, so in the morning class, we have seen this particular picture. We have talked about the outbreeding devices. We have discussed the pollen crystal interaction. We have also seen artificial hybridization. So these are the topic which we have covered, right, in the morning's class. Along with that, we have also understood what is actually happening inside the ovule. So ovule में आपका वहाँ पे क्या हो रहा है? That is also something we have understood to a certain extent. We have tried to understand that as well. So we know that inside the ovule, you will be having your antipodal cell, you have your pollen nuclei, you have your syndicate cell. But once the pollen tube has to enter inside the ovule, जब मेरे pollen tube को ovule के अंदर आना है, then one of the syndicate cell, both the syndicate cell will undergo will be like dying over there. They will degenerate. And once my syndicate cell die over there or degenerates, then you have a free pathway over there. You have a free pathway through which you can enter inside. Right? Something we have seen that. Also, we have discussed that a pollen tube is always going to have two male gametes. Your pollen tube is always going to have a two male gametes, and those two male gametes will be released in the embryo sac. वो आपके embryo sac में हम यहाँ पे release कर देंगे, right? Now, once the two male gametes come over here, after that we have very categorically seen that one of the male gamete will go and fuse with the two polar nuclei, and now you know these polar nuclei are my n plus n, right? These are my two haploid nuclei over there. And now my male gamete comes and attaches itself with the two polar nuclei, and that will lead to a formation of my triploid nucleus, right? Triploid endosperm. So this triploid structure which has been formed over here, this triploid structure which has been formed over here, this triploid structure is called as my primary endosperm nucleus. So that is something which has been formed. ये मेरा यहाँ पे बन जाता है. Okay, so that is something you'll be finding forming over here. So that is, you can also collectively use a single term. It can be also called as my endosperm, which has been formed right now. Okay. Now endosperm is something which is going to nourish the embryo. Please know this thing very carefully, and you have to remember this thing very clearly, that embryo is something whose role is going to be to nourish the developing embryo. Endosperm ka role hoga to nourish the developing embryo, right? So that is something you find happening. And along with this, there is one more thing which is happening over here. That is your male gamete fusing with the egg. Your male gamete coming, fusing with the egg. That is my haploid egg. और जैसे ही इसके साथ fuse करेगा, that will lead to a formation of my zygote. That will lead to a formation of a zygote, and that same zygote will develop with the course of time into your embryo. The same zygote will develop in a course of time into your embryo. So that is something you have to know. That male gamete fuses the egg, converts into, forms into a zygote, and now this zygote is something which will develop into your embryo. So this is what you will be finding happening here. Okay. Now understand one thing very carefully. Whenever the male gamete is going to fuse with the egg, this event is called as my syngamy. This event is called as my syngamy. So syngamy simply means that when my male gamete is going to fuse with the egg, that will lead to a formation of a zygote, and that event over here will be remembered as a syngamy. That event is called as my syngamy. तो एक तो ये बात दिमाग में बिठा लो, right? That is something you have to know. उसके बाद come back over here. You are finding a male gamete combining with my two polar nuclei. My male gamete over here combining with my two polar nuclei. This event will be called as my triple fusion. This event has a term, and this event is called as my triple fusion. ठीक है? This event is called as my triple fusion. I hope you can understand why it is called my triple fusion, because you have a three haploid nuclei actually combining with each other. So this event over here is called as my triple fusion. Okay. And collectively, they are a single term. Collectively, you can see that one male gamete is fusing with the polar nuclei, other male gamete is fusing with your particular egg. So that is why this event is also called as my, collectively, these two events over here are called as my double fertilization. Collectively, these two events are called as my double fertilization. Now, when you get a term of a double fertilization, then always remember one thing very carefully over here, that it simply means that one male gamete fusing with the egg, other male gamete fusing with the polar nuclei. That is going to be called as my double fertilization. Now, there's one thing you should be very clear with, that double fertilization is something which is only peculiarly seen in my angiosperms. Peculiarly, you will be finding them happening in the angiosperm. You don't find something like this happening in my जिम्नोस्पर्म्स. ऐसा कुछ नहीं होता वहां पे. जो भी आपका यहाँ पे हो रहा है वो आपका हो रहा है एंजियोस्पर्म के अंदर. ये बात आपको यहाँ पे याद रखनी है. That's a first separate thing you should be clear with. What do you get after double fertilization? तो याद रखना, डबल fertilization के बाद आपको मिलता है zygote, and second thing you'll be getting is going to be your endosperm. और ये एंडोस्पर्म बनाना is going to be a very important event, because endosperm is always going to nourish your developing zygote, which will turn into the embryo. So yaad rakhna, endosperm is a nutritive material, right? It is a food storage bag. It will have high amount of nutrition, and that nutritive material is something used by the developing zygote as a source of food. So isko aap use karte ho as a source of food. So please remember, the endosperm will be used as a source of food, by embryo. So embryo is actually going to develop eating this endosperm. And please carefully all remember that endosperm is always going to be triploid in nature. It is always going to be triploid in nature. And zygote क्या होगा मेरा यहाँ पे? Zygote क्या होगा?
Diploid.
राइट, zygote मेरा यहाँ पे रहेगा डिप्लॉयड. zygote मेरा यहाँ पे डिप्लॉयड रहेगा. ठीक है, इतनी कहानी है, बड़ी आसान सी कहानी है, ज्यादा कुछ इसमें कॉम्प्लेक्स है नहीं. इतनी बातें आपको यहाँ पे याद रखनी है. व्हाट इज माय ट्रिपल फ्यूजन, व्हाट इज माय सिंगैमी, डबल फर्टिलाइजेशन होने के बाद आपको क्या मिल रहा है, एंड व्हाट इटसेल्फ डबल फर्टिलाइजेशन एक्चुअली मींस. सो दिस इज व्हाट यू हैव टू नो इन टर्म्स ऑफ दिस. नाउ आई हैव अ क्वेश्चन. माय क्वेश्चन इज, क्वेश्चन ध्यान से सुनना. क्वेश्चन कुछ ऐसा है दैट you have a let's say you have a male plant. And male plant is tetraploid. Tetraploid means it is 4n structure, right? And then you have a female plant, and female plant is going to be diploid. So you have a tetraploid male plant, you have a female plant which is a diploid plant. You tell me how the endosperm and zygote are going to look in this plant. Take your time, अपना टाइम ले लो और सोच के बताओ कि इसमें एंडोस्पर्म कितने प्लॉइडी का होगा, how the endosperm is going to be in them, and how the zygote is going to be in them. So now take your time and tell me. बड़ा ही आसान सा है, आराम से सोचो, कर सकते हो, कोई बहुत कॉम्प्लेक्स सवाल नहीं है, थोड़ा सा सोचना पड़ेगा और हो जाएगा ये. You have to tell me what is my endosperm is going to be made up of and what is my zygote is going to be made up of. चलिए स्टार्ट करिए करना, and then raise your hand once you're done with this. I hope क्वेश्चन चारों को आपको यहाँ पे क्लियर है. I hope question is clear to all of you. Male plant is 4. हाँ, क्वेश्चन क्लियर है ना? बस now try to solve this. हिंट चाहिए थोड़ी या कर लोगे खुद से? लगता है खुद से कर लेंगे. काफी टाइम लग गया। तनिष्क हो रहा है या थोड़ी हिंट चाहिए? गायत्री, शिखा एंड मिसबा, बाकी लोग हेल्प चाहिए या हो कर लोगे? व्हाट इज हैपनिंग? कोई कुछ बोल ही नहीं रहा। ऑल ऑफ यू आर लाइक की
So will the structure be tetraploid?
क्या एंडोस्पर्म आप क्या कह रहे हो? टेट्राप्लॉइड?
Yes, sir.
सो आप कह रहे हो मुझे एंडोस्पर्म आपका होगा टेट्राप्लॉइड होगा, और जाइगोट? बताइए शिखा, बताइए जाइगोट। वापस बोलना, आवाज कट गई आपकी। तनिष्क, नेटवर्क इशू है क्या? कुछ दिक्कत आ रही है? वापस ट्राई करो, बोलो। अच्छा, अरे बाबा, शिखा क्या होगा? जाइगोट जल्दी बोलो, चलो अभी।
Tetraploid.
जाइगोट भी टेट्राप्लॉइड, एंडोस्पर्म भी टेट्राप्लॉइड। जाइगोट आप कह रहे हो कि 2n होगा। ये देखो किसी का आ रहा है 2n। और बाकी गायत्री एंड मिसबा, व्हाट यू हैव?
Zygote 2 diploid.
जाइगोट 2n। ठीक है, चलो मैं ही करता हूँ इसे। चलो अब मैं कर रहा हूँ यहाँ पे, से ध्यान से देखो। ये वो मेल प्लांट, मेल प्लांट इज गोइंग टू बी 4n प्लांट। ठीक है, मेल प्लांट मेरे यहाँ पे है 4n, राइट? अगर मेरा मेल प्लांट 4n है, राइट, so that means it is my, even इसके megaspore, sorry, microspore mother cell are also going to be 4n. If male plant is 4n, then I hope you understand that in the male plant you will have a MMC, microspore mother cell, that is also going to be 4n only, right? I hope कोई confusion नहीं है, confusion हो तो पूछ लेना interrupt करके. It is also going to be 4n over here, right? Now you know that this microspore mother cell will undergo the meiosis, will undergo meiosis. Now if it undergoes meiosis, how many cells I will get? How many cells you get after a microspore mother cell undergoes meiosis? Meiosis करेगा तो मुझे कितने सेल मिलेंगे यहाँ पे? अरे 4 सेल ना। अरे इट इज अंडरगोइंग meiosis बाबा, इट इज अंडरगोइंग meiosis. अगर ये meiosis करेगा तो मुझे मेरे 4 सेल मिलेंगे यहाँ पे. I will always have my 4 cells will be formed over here, right? And all those 4 cells are going to be 2n cells. हाफ हो जाता है ना बेटा, जब आपका meiosis होता है तो आपका नंबर हाफ हो जाता है. जब डिप्लॉयड होता था आपका माइक्रोस्पोर मदर सेल, यूज्ड टू गेट अ हैप्लॉइड माइक्रोस्पोर, बट हियर because it's a tetraploid, so all the microspore which have been formed are going to be 2n. So we'll have a half the number of the chromosome will be there in the microspore. क्या इतनी बात समझ आ रही है आपको यहाँ पे? Is this clear to all of you?
Yes, sir.
Tanishk and other people, Misbah and Gayatri. Okay, so these are my microspores which have been formed. Now if my microspore is 2n, so can I directly say that if they are 2n, then obviously my male gametes which are going to be formed inside them, they are also going to be 2n. So जो मेरे male gametes जिसके अंदर बनेंगे वो मेरे 2n ही होंगे obviously. क्या इतनी बात क्लियर है आप सबको?
Yes, sir.
Give me a thumbs up अगर clear है तो. Give me a quick thumbs up if it is something clear to you. Right? Okay. So that is something we'll get to see. So all my male gametes are also going to be 2n over here. Okay. So now you know that my male gamete is going to be 2n. Male gamete is going to be 2n over here, right? Now let's talk about my female plant. Female plant अगर मेरा diploid है, so all the cells in the embryo sac are going to be haploid. All the cells in the embryo sac, so पूरा के पूरा जो मेरा embryo sac होगा, the complete embryo sac which I'll be finding, the embryo sac is going to be what? Is going to be diploid, is going to be haploid. अगर मेरा फीमेल प्लांट डिप्लॉयड है तो मेरा पूरा एम्ब्रियो सैक मेरा हैप्लॉइड होगा. क्या ये बात सबको यहाँ पे समझ आ रही है? पहले ये बताओ. तनिष्क, गायत्री, शिखा, मिसबा, आई होप इट्स क्लियर टू ऑल ऑफ यू.
यस सर.
ठीक है, कोई कंफ्यूजन यहाँ पे होना चाहिए कि ये सारा मेरा यहाँ पे हैप्लॉइड होगा, राइट? अब तुम सोचो यहाँ पे, यू हैव अ हैप्लॉइड एम्ब्रियो सैक. If you have a haploid embryo sac, now let's make two polar nuclei. Let's make two polar nuclei. So your polar nuclei will like my N, and you will have one more polar nuclei, that's going to be N, right? अब जो मेल गैमेट इससे आके फ्यूज कर रहा है वो है मेरा यहाँ पे डिप्लॉइड. द मेल गैमेट which is coming and fusing over here, that male gamete is going to be diploid. So that male gamete over here is going to be diploid. So if I have a male gamete, and if the male gamete is going to be diploid, then now count over here. 2n plus n plus n कितना हो रहा है?
4.
Right, 4n. So that is why I will clearly say over here that my endosperm is going to be my 4n. So over here what will happen? My endosperm is going to be 4n. So my endosperm here is going to be 4n. That's what Shika said, and that's going to be a true thing. So that is what I'll be finding over here. Endosperm is going to be 4n over here, right? And then let's talk about zygote over here. अब zygote के साथ यहाँ पे क्या होगा? That zygote is going to be your what? Zygote के अंदर egg is going to be my haploid egg. Is going to be haploid egg, and now the male gamete is going to come. Male gamete is going to be your 2n. Male gamete is also going to be a 2n over here. So if a 2n combines with the n over here, then my zygote is going to be what? क्या होगा मेरा zygote? Triploid.
3.
Yes, 3n. So that will be my zygote. So no matter how much complex question they are going to ask you, you should be able to answer them. आसान नहीं है, ठीक है? You just have to do the half of the male plant. Male plant में अगर बोला गया है कि आपके इतने number of chromosomes है या you have 4n, 6n जो भी है, you have to do the half of that, right? Similar goes for the female plant as well, right? And then you just have to fuse the gametes, and you should be able to get the answer. So endosperm is going to be my 4n over here, and the zygote is going to be my 3n. So that should be clear to all of you. All right. चलो आगे बढ़ते हैं. You will find such question again. I will give you such question again. So you should be able to like solve them all the time. So let's see what will be seen. So now this is something we have done. We have when we talk about the fertilization, we make the male gamete, right? Similarly, we make the female gamete. Okay, that's going to be a female egg. And then what happens? You will find the zygote has been formed. Male gamete, female gamete fuse, and the zygote is getting formed. You should also know one thing, that one male gamete is going to fuse with the one female egg, and that will lead to formation of a zygote, right? On the other hand, another male gamete, second male gamete is going to fuse with the polar nuclei and will give me the endosperm. Will give me the primary endosperm nucleus, right? 3n. So that is nothing but my endosperm. So that should be clear to all of you. Okay. Now, let's see over here. So double fertilization, after entering one of the synergid cell, the pollen tube bursts and releases the two male gametes. Yaad rakhna, the synergid cell is going to die, right? And pollen tube is going to release your two male gamete, will be released by my pollen grain. And then what happens? One of the male gamete moves towards the egg and fuse with the nucleus and completing the syngamy. So when it fuses with the egg cell, it is called as my syngamy, something I have told you. Result, it's into what? A diploid cell or a zygote is been formed. That will lead to formation of a zygote, and zygote is also going to be my 2n structure. Right? So male gamete plus egg, syngamy, and that will lead to formation of my 2n zygote. Okay, so you can see this happening. So you have that is my syngamy. Okay, then male gamete plus secondary nucleus. Now, what is my secondary nucleus? This is nothing but my polar nuclei. Secondary nucleus is nothing but my tension मतलब नया term है. It is nothing but my polar nuclei. It is going to be your polar nuclei. So my male gamete fused with the secondary nucleus and will give rise to your primary endosperm nucleus. Since two types of fusion are taking place, syngamy and triple fusion takes place in the embryo sac. This phenomena is called as my double fertilization. Because these two events are taking place in the embryo sac, you are going to call it as a double fertilization. So ये आपको clear होना चाहिए. And याद रखना, primary endosperm nucleus is going to be triploid, right? This event only occurs in the flowering plants. That means angiosperms के अंदर ही आपको ये होते हुए आपको मिलेगा. Okay. Now let me discuss few things about the endosperm. Let me just discuss few things about the endosperm. So before that, yeah, just यहाँ पे. Okay. Now, what will happen now? What generally you will get to see that you have your haploid cells, haploid polar nuclei. you have your male gamete. You have your male gamete, haploid 1. They will come and fuse over there. And once the fusion happens, this will turn into the endosperm. This will turn into the triploid endosperm. Right? So now this is my 3N endosperm. It is called as my primary endosperm nucleus, PEN. That is something it will be called as. It has been formed. अब यहाँ पे क्या होता है? Now what we have generally observed that once this nucleus has been formed, once this endosperm nucleus has been formed, it will undergo rapid mitosis. Or what will happen? A nuclear division or nuclear mitosis will be seen happening over there. Okay. Once this primary endosperm nucleus has been formed, what will happen? We will find multiple times nuclear division will be seen. Multiple nuclear division will be seen. Nuclear division is seen. Division is seen. So that means what? This nucleus will undergo rapid division. Primary endosperm nucleus will undergo rapid division. Okay. And will increase in number without any wall formation. Without any wall formation. So the primary endosperm nucleus you have now, it will undergo multiple nuclear division without wall formation. So what will happen because of that? You will find a large number of nucleus will be formed. A large number of nucleus will be seen over there. There we go. So what do you get to see because of that? What do you get to see because of that? You will find a large number of nuclear endosperm will be seen. इसने multiple times इस nucleus ने किया mitosis. The nucleus has undergone multiple times mitosis over there, and that is why you have a large number of nucleus can be seen, which have been formed. And such kind of endosperm, where you have a large number of nucleus has undergone mitosis and a large number of like nuclear after nuclear mitosis, a large number of nucleus will be seen over there. This is called as my free nuclear endosperm. This is called as my free nuclear endosperm. This is called as your free nuclear endosperm. Now you have to know one thing very carefully over here. That is free nuclear endosperm which has been formed now. You can clearly see in the coconut water. So the coconut water जो तुम पी रहे होते हो, the coconut water which you are drinking, the liquid is nothing but my free nuclear endosperm. That is your free nuclear endosperm. So that is something you have to know. Okay. So this nucleus will undergo, primary endosperm nucleus will undergo multiple time nuclear division to give me a free nuclear endosperm. And once a large amount of nuclear endosperm has been formed, after that the wall formation can be seen. After this has been formed, now the wall formation can be seen. So after this you can find later on the wall formation can be seen. So एक बार nuclear division बहुत सारा हो गया, then you will find wall formation can be observed. So after this, the wall formation will be seen. Wall formation is seen afterwards. So that should be clear to all of you. That wall formation will be happening later. Okay. Initially, primary endosperm nucleus will undergo rapid mitosis, will increase its number, and then later on the wall formation will be observed. And when this primary endosperm nucleus will undergo rapid mitosis, whatever the endosperm has been formed is called as my free nuclear endosperm. That is called as my free nuclear endosperm. And the best example of my free nuclear endosperm is going to be the coconut water. जो तुम पी रहे होते हो. That's the example of my free endosperm nucleus. ठीक है? Free nuclear endosperm. So क्या इतनी बात आपको यहाँ पे समझ आ रही है? Is this thing clear to all of you? All four of you? Right? तनिष यहाँ तक clear है? तनिष, you can just अभी तुम्हारे यहाँ पे network issue है ना, so you can just give me a thumbs up, तब भी चलेगा. Okay. So ये आपको यहाँ पे होते हुए इस तरीके से आपको यहाँ पे मिल सकता है. You can find this happening over here. Right? So you should know what is my PN and what will be the events happening with the PN, and it is all going to be my triploid structure. Right? Now there's also one thing I want to tell you over here, which you get to see in the within the ovule, that endosperm is going to be like highly nutritive structure. It is something which is going to nourish the embryo. Always remember the development of the endosperm will happen faster than the development of the embryo. So development of the endosperm development of endosperm precedes, someone has a question, can no, precedes the embryo growth. That means what? That endosperm is going to develop rapidly and it will develop faster than the growth of the embryo. Or rather, I can say that the endosperm will develop rapidly and there will be the growth of embryo will happen very slowly or will happen after certain growth of the endosperm. अब ऐसा क्यों हो रहा है? क्यों आपका एंडोस्पर्म पहले बनेगा ज्यादा और फिर आपका एंडोस्पर्म एम्ब्रियो आपका डेवलप करेगा? The reason is that you have to nourish the developing embryo. You have to nourish the developing embryo, and that is why the endosperm development will happen faster or will happen much earlier than the growth of the embryo. So आपके embryo के growth के पहले ही आपका endosperm बन के ready हो जाएगा. ये बात याद रखना, that development of endosperm is going to precede the growth of the embryo. So that is one thing you have to know. ये एक चीज आपको यहाँ पे याद रखनी है. This should be clear to all of you. Okay. Now, what do you generally get to see now? When you observe the endosperm, it's a 3N structure. It is going to be a triploid structure. You know that. The outer layer of the endosperm is called as my aileron layer. The outer layer of endosperm is called as my aileron layer. Now, this aileron layer is rich in protein. If they ask you what is the outer layer of endosperm, you should have to tell them it's my aileron layer, and it is going to be rich in protein. That's one thing you have to know. And what kind of content you have in the endosperm, inside the endosperm, then you can tell them endosperm is generally going to have a huge amount of my starch. Endosperm is generally going to have a huge amount of starch. So, आपको बीच में आपको यहाँ पे मिल रहा है inside the endosperm. So, endosperm will be having a huge amount of your starch. So, starch content will be very high inside the endosperm, and the outer layer of the endosperm is called as my aileron layer, and aileron layer is going to be rich in protein, right? And every structure over here is my triploid. Every structure here is going to be my triploid in nature. तो इतनी बात आपको यहाँ पे याद रखनी है. This is one more thing which you should be very much clear with, right? Now, development of the endosperm and embryo, maturation of ovule into the seeds, ovary into the fruit are collectively termed as a post-fertilization event. So, after fertilization, double fertilization, what will happen? The ovule will start developing into the seed. Ovule will start turning into a seed over here. That will turn into the seed, right? Your ovary will turn into the fruit. That will also initiate. आपका ovary will fruit में convert हो जाएगा. Okay, and these are called as my post-fertilization events. You call them as a post-fertilization events. Now let's see some things over there. So you can see your ovule will turn into the seed and the ovary will turn into the fruit. चलो आगे देखते हैं. So now let's see what they have written about the endosperm. It's a product of triple fusion and develops from a central cell of the embryo sac, and will develop from the central cell of the embryo sac, right? It is generally going to be triploid tissue, something we know that. The cells of the tissue are filled with the reserve food material. याद रखना, they are filled with the reserve food material. Outer layer को हम क्या बोलते हैं? What is the outer layer is called as?
Aileron.
Aileron.
Right. It's called as my aileron layer. And it is going to be rich in my protein. So that should be clear to all of you. Okay, that is my endosperm. They are used for the nutrients of the developing embryo. So basically, they are something nourishment जो आपको मिलता है ना यहाँ पे embryo को, that is been coming from the endosperm. Okay. Now, mature seeds are of my. So now you know the basic idea about the endosperm is clear to you. Now, mature seeds are of my two types. Mature seeds आपके यहाँ पे होते हैं दो type के. Let's see them. So what will happen now when the embryo is growing inside the seed? जब आपका एम्ब्रियो सीड के अंदर ग्रो कर रहा है, आपकी दो चीजें यहाँ पे हो सकती है. देयर इज अ पॉसिबिलिटी दैट द एम्ब्रियो कंप्लीटली कंज्यूम्स ऑल द एंडोस्पर्म. जो भी एंडोस्पर्म है सीड के अंदर वो सारा वो कंज्यूम कर जाए, सारा खा जाए. दैट्स अ वन पॉसिबिलिटी. दूसरी पॉसिबिलिटी आपकी क्या है कि आफ्टर मैच्योरेशन ऑफ द एम्ब्रियो यू स्टिल हैव सम एंडोस्पर्म देयर इन द सीड. आफ्टर द डेवलपमेंट ऑफ द एम्ब्रियो यू स्टिल हैव small amount of endosperm still present in the seed. तो दो चांसेस है आपके यहाँ पे, दो पॉसिबिलिटी आपको मिल सकती है सीड के अंदर. You have one type of seed in which the complete endosperm is getting consumed. You have one another type of my seed where you will find only small amount of endosperm still remains after the consumption. सारा खाने के बाद कुछ अभी भी वहां पे आपका रह रहा है. ठीक है. So on the basis of that you have two type of seeds. On the basis of that you have two type of seeds. So let's try to see that. You have non endospermic seed. नाम से पता चल रहा है नॉन एंडोस्पर्मिक सीड. यहाँ पे आपका सब कुछ आपने कंज्यूम कर लिया होगा. दे आर ऑल्सो कॉल्ड एज माय एक्स एल्ब्यूमिनस सीड. सो व्हाट विल हैपन इन सीड लाइक माय ग्राम पीएन ग्राउंडनट. ये एग्जांपल याद रखना, ये क्वेश्चन आता है एग्जाम में. इन द एग्जांपल ऑफ इन द सीड्स लाइक माय ग्राम पीएन ग्राउंडनट the endosperm is completely consumed. पूरा खा लिया जाता है by the embryo. Thus the seed are called as my non endospermic or X albuminous seed. और आप यहाँ पे देख लो अपनी पिक्चर, यहाँ पे ग्राम पी और ग्राउंडनट की पिक्चर आप यहाँ पे देख लो. राइट. एग्जाम्पल याद रखने आपने सबने ये सारे ग्राम पी एंड ग्राउंडनट आर एग्जाम्पल ऑफ माय नॉन एंडोस्पर्मिक और एक्स एल्ब्यूमिनस सीड. यहाँ पे आपका एंडोस्पर्म पूरा आप यहाँ पे कंज्यूम कर लिया जाएगा. कोई कंफ्यूजन हो रहा हो तो हैंड रेज करके पूछ लो. इफ यू हैव एनी काइंड ऑफ कंफ्यूजन देन प्लीज रेज योर हैंड एंड आस्क योर क्वेश्चन. ठीक है. सो दैट इज माय नॉन एंडोस्पर्मिक सीड. राइट. ओके, अब आगे देखते हैं. देन यू हैव अनदर कैटेगरी. व्हाट वाज अनदर कैटेगरी? बताओ, कोई मुझे बताओ. मिसबा क्या थी दूसरी कैटेगरी मेरी यहाँ पे? क्या थी? ये तो क्या नॉन.
Albuminous seed.
बोलो मिसबा.
Albuminous seed.
उसमें क्या होता है? व्हाट इज देयर इन द एल्ब्यूमिनस सीड?
Endosperm is present.
Yes, you will find even after the growth of the embryo, development of the embryo, you will find small amount of endosperm can be seen in them, and that is my albuminous, or you can call him as an endospermic seed. So let's have a look at them. So you have an endospermic or albuminous seed over here. In the monocot, like castor and bean, embryo does not consume all the endosperm, so it persists in the mature seed. So mature seed में आपको endosperm आपको मिलेगा देखने के लिए. Such seeds are called as my endospermic or albuminous seed. I think it's a very simple concept. You just have to categorize them in your head. दो कैटेगरी के अंदर. Right? देख लो यहाँ पे wheat, maize. तो बहुत सारे एग्जाम्पल है आपके यहाँ पे. या वो wheat, या वो maize. wheat मतलब गेहूं आपका, maize मतलब आपका गन्ना. Not गन्ना, maize मतलब your corn. Right? Then you have a barley, sunflower and coconut. Okay, these are all going to be the example of my endospermic seed. Right? कोकोनट के अंदर आपको पूछा जाएगा द कोकोनट वाटर इज एग्जांपल ऑफ माय फ्री न्यूक्लियर एंडोस्पर्म. दैट्स एग्जांपल ऑफ माय फ्री न्यूक्लियर एंडोस्पर्म. राइट. So you can see you have a wheat, you have a maize, and then you can see a picture of a sunflower seed, barley, and your coconut. So they are going to be the example of my albuminous or my endospermic seed. Okay, so ये हमने यहाँ पे देख लिया. Now let's discuss something in terms of the embryo. So now we are done with the endosperm. Now we'll focus on the embryo. Now we'll try to understand what has been there in the embryo. Right. Now when we discuss the embryo, the development of the embryo will happen from the zygote, and that is called as my embryogeny. Right. तो आपके zygote में से ही आपका embryo बनता है. So याद रखना, initially the zygote has been formed, and from that zygote, the embryo is getting formed. Embryo will be much more larger than the zygote. हमेशा रखना, embryo will be much more larger than the zygote, and zygote will form the embryo via cell division, and that cell division is called as my mitosis. हमेशा रखना, ये जो आपका ग्रोथ हो रहा है ये all happening because of the mitosis. Meiosis नहीं होगा, हो गया जो होना था वो. Because of the mitosis, the embryos will be formed, and this embryo will just keep on growing over there. When I say the embryo is growing now, that actually means what? कि वहाँ पे cells add हो रहे हैं. When I say the embryo is growing, that actually means what? कि वहाँ पे आपके cell add किए जाएंगे. So the cells are getting added over there. And when I say the cells are getting added, that itself means कि वहाँ पे आपका mitosis की वजह से आपके cell आपके यहाँ पे add किए जाएंगे. The mitosis is the reason because of that the cells are getting added over there. So embryo is also going to be diploid. Zygote भी आपके यहाँ पे diploid ही था. Okay. Now let's try to see a few things. You have an embryo develops at the macropyle end of the embryo sac where the zygote is getting situated. Most zygote divide only after a certain amount of endosperm is formed. So something I've just told you, that most zygote will only divide once a certain amount of endosperm is getting formed. That's what I've told you. Endosperm growth will happen much more rapidly compared to the zygote. क्योंकि आपको feed करना है. Because you तो ऐसा नहीं है कि आप रैबिट लाओगे फिर आप लेने के लिए जाओगे उसका खाना, पहले आप उसका खाना लेके आओगे देन आप रैबिट लेके आओगे, राइट? सिमिलरली यहाँ पे आप पहले खाना बनाते हो and then the zygote is going to develop over here. Okay, now let me tell you a couple of things about the embryo. Now when we discuss, let me just, yeah, I do have a space. Now what happens now when you discuss the embryo development, when you discuss the embryo development, there are a few things which you have to remember. First of all, the first thing which you have to know here is, just wait a minute, the first thing you have to know that it is all happening because of the mitosis. It is all happening because of the mitosis. ये पहली बात आपको यहाँ पे याद रखनी है. Now when the embryo is going to develop, how the growth is going to be. Now the development of the embryo is a very detailed process, but हम उसे इतना detail में नहीं सीखेंगे. We won't understand that process in detail. The reason is because you don't find questions coming from that. It is not something which has been seen to be coming as a part of my NEET exam. You don't get a detailed question. You just have to know some basic things, right? So like initially you have a single-celled zygote. You have a single-celled zygote. It will become two-celled, and then it will just keep on growing. Right? It will become two-cell, and then it will just keep on growing. So it is my zygote, and will divide, and will just keep on growing over there, right? So I'm directly going to go on one of the stages over there. So when your embryo develops, when the embryo is going to develop, so it will just, the number of cells are going to keep on increasing, right? Number of cells are going to keep on increasing, and then you will have a stage which is called as my globular stage. This will just keep on growing, and then you will have a stage called as my globular stage. Globular stage. From this globular stage, the embryo is going to again grow much more in this, and it will become heart-shaped. It will become heart-shaped. So after this, the embryo is going to grow much more in detail, and it will become something like this. याद रखना ये काफी डिटेल प्रोसेस है, लेकिन हम उसे डिटेल में नहीं सीख रहे because you don't find question coming from this. That's why we won't be wasting time on this. But then you will have a globular stage and you have a heart-shaped stage. You have a heart-shaped stage. Then this will grow further. It will look something like this now. It will become something else. Such stage over here, this stage is called as my torpedo stage. This stage over here is called as my torpedo stage. ये torpedo पता है क्या क्या होता है? Anyone knows what is torpedo? No one. अच्छा, मैं आपको थोड़ा बताता हूँ. आपने शायद देखा होगा पिक्चर के अंदर. पता है सबमरीन के अंदर, आपने कभी सबमरीन की कोई पिक्चर देखी हो या नेट जियो पे देखा हो, तो सबमरीन के अंदर क्या होता है, एक वो मिसाइल जो लोग रिलीज करते हैं सबमरीन के अंदर, पानी के अंदर, वो आपकी जो जा रही होती है जाके शिप को हिट करती है. दैट मिसाइल इज कॉल्ड एज माय टॉर्पेडो, राइट? ये टॉर्पेडो कई बार आप प्लेन से भी फेंक सकते हो अगर आपको किसी शिप को हिट करना हो. तो बेसिकली शिप को डैमेज करने के लिए जो आप मिसाइल यूज करते हो उसको हम कहते हैं टॉर्पेडो. यू माइट हैव सीन देम or might not, but yeah, that is called as my torpedo. So it looks like a torpedo. Okay, so याद रखना, ये सारे स्टेज के नाम याद रखना. That you have a globular stage, then you have a heart-shaped stage, and then you have a torpedo stage. और उसके बाद you will have a much more mature stage. Mature stage में आपको क्या मिलेगा? That you can see a two cotyledons are very clearly seen. So you can see in this stage, two cotyledon will be very clearly seen over here. So दो cotyledon आपके यहाँ पे काफी clearly आपको मिल जाता है. So दो cotyledons, you can see these two cotyledons will be seen. Cotyledons. Now, what kind of question they can ask you? They can ask you question like, you have to arrange these stages. So you have to know, first is my globular stage, then heart stage, then torpedo stage, and then you have your two cotyledons or mature stage has been seen. That's my mature embryo, right? So they might ask you to arrange them. So याद रखना sequence क्या रहेगा, single celled, two celled, और भी बीच में stage होता है, but you have to know globular, heart stage, torpedo, and then you have the mature stage. This is my mature embryo. That is your mature embryo. And a mature embryo will have two leaf-like structures, and these leaf-like structures are called as my cotyledon. So this it should be clear to all of you. क्या इतनी बात आप सबको यहाँ पे समझ आ रही है? गायत्री, मिसबा, तनिष, शिखा, is it clear to all of you? All right. Okay. ठीक है, now let's move ahead. So there is something you will get to see. So this is adaptation to provide the assured nutrition to the developing embryo. That means you make an embryo endosperm grow earlier than the embryo. That is the adaptation, right? Now it means endosperm development precedes the embryo development. Something I've already told you, that endosperm development is going to precede the embryo development. You know this. So early stage of embryogeny are similar in both monocots and dicots. Monocot and dicot both will have early stage, are going to be similar. So I can show you a picture over here. So you can see you have a single celled zygote, then it is going to grow over there, and then you can see all the stages over there, right? You can see all the stages. You can see a globular stage. Remember this one. Remember the heart-shaped stage. Then you have a torpedo stage, and then you have the mature embryo. And the mature embryo, you can very clearly see in the mature embryo, you have a presence of my 2 cotyledons can be seen over there. ठीक है, 2 cotyledon मुझे यहाँ पे clearly मुझे यहाँ पे देखने के लिए मुझे यहाँ पे मिल जाता है, right? तो इस stages के नाम याद रखना. These are stages in which the embryo is going to grow. ठीक है, क्या इतनी बात आप सबको यहाँ पे समझ आ रही है? Is this thing clear to all of you? अच्छा, I'm not getting any answer. अच्छा, now I can see some thumbs up. मिसबा, शिखा एंड तनिष, पता चल रहा है यहाँ पे आपको?
Yes, sir.
All right, okay. Just wait a minute. तनिष कहाँ गया हमारा? Okay, ठीक है, चलो. All right. Now, so that is something now we have seen. Now let's try to talk about something in terms of my dicot embryo. So dicot embryo की हम यहाँ पे specific बात करें. So let's discuss something in terms of my dicot embryo. Now when you discuss the dicot embryo, there are few things you have to know. Dicot means you will have presence of 2 cotyledons. You will have presence of 2 cotyledons. Now let me make a simple structure in front of you. एक आपके सामने एक मैं simple सा structure बनाता हूँ, just to make you understand this thing very in a simple way. Assume that this is my embryonal axis. This is my embryonal axis, right? This is my embryonal axis over here. और ये center है मेरे यहाँ पे. This is the center point. अब center point से you have your cotyledons are coming out. You have one cotyledon which is coming out from here, right? And you have another cotyledon coming out from here, from the center. So dicot था मेरा embryo यहाँ पे, so 2 मैंने यहाँ पे cotyledon मेरे यहाँ पे है. So these leaf-like structures are called as my cotyledons. Okay. If someone asks you what they do, they provide the nourishment to the developing embryo. So they also provide the nourishment. They're also going to provide the nourishment to developing embryo. इनका काम होता है nourishment provide करना मेरे developing embryo को. Right? Now pay attention over here. These are the midpoint over here. This is the middle region, and this is where the point from where I am finding the cotyledons are coming out. Okay. Now what do you get to see in the embryo? Na? The upper part over here, this upper part is called as my plumule. This upper part over here is called as my plumule. Right? Similarly, you will have a lower region. The lower region over here is called as my radicle. The lower region over here is called as my radicle. याद रखना, radicle is a site from where I'll find my root system is going to come out. Radicle is a site from where I'll get to see my root system is going to come out. So यहाँ से मेरा निकल के आएगा मेरा यहाँ पे root system. ठीक है, तो पूरा जो root development आपका यहाँ पे होता है, the root development you will get to see. That root development is something which will be happening from your particular this thing, from my radical region. On the other hand, you have the plumule part. The plumule is well known for formation of my shoot system. So plumule is an area which is well known for my formation of my shoot system. So याद रखना, जो मेरा plumule यहाँ पे होगा, ये आपको बना के देगा आपका यहाँ पे. मैं इसको यहाँ पे लिखता हूँ. It will form my shoot system. It will form my shoot system. Is this thing clear to all of you? क्या इतनी बात आपको यहाँ पे समझ आ रही है? Is this thing clear to all of you?
Yes, sir.
ठीक है, और बाकी लड़कों को भी आई होप ये चीजें यहाँ पे समझ आ रही है. So you're raising your hand. There's some issue with my phone. I guess I cannot see you, all of you. Okay, chalo. So that is one thing you have to know. The second thing what you have to understand here is, now you have a region, you have a small region over here between the point from where the cotyledons are coming out and between the plumule. So plumule और इसके बीच में जो आपको यहाँ पे area दिख रहा है, let me mark this area. ये जो आपको area दिख रहा है between the point of cotyledon coming out. Sir, so today I am having some very slow internet. हाँ, आज कुछ लग रहा है, कुछ concern है. दिल्ली में भी कभी कभी ये कंसर्न आ सकता है, कोई नहीं, ठीक है. ऑलराइट, so from here if you can see this area between the point of attachment of the cotyledon and my plumule, इसके बीच में ये जो एरिया है आपका, this area is called as my epicotyle. This area over here is called as my epicotyle. ठीक है. The point from where the cotyledons are coming out and the region above that is called as my epicotyle. So a region above my point of attachment of my cotyledons and my plumule, इसके बीच में जो एरिया मेरा यहाँ पे होता है, इसे हम बोलते हैं यहाँ पे as a epicotyle. Similarly, you have a region below that, the point from where the cotyledons are coming out and between my radicle. So इसके बीच में भी आपको यहाँ पे एक एरिया मिल रहा है, and that area here is called as my hypocotyle. Hypocotyle. तो ये चीज आपको यहाँ पे याद रखनी है. So two terms has to be remembered over here. What is my epicotyle and what is my hypocotyle? So epicotyle is a region between my plumule and the point of attachment with the cotyledons. That region over here in the embryonal axis is called as my epicotyle. And similarly, you will have a hypocotyle in the lower part. In the lower part between the radicle and the point of attachment of the cotyledon is called as my hypocotyle. So these are the two things which you have to remember. So these are the 2 points which you have to know. So you have a hypocotyle and you have a epicotyle over here. ठीक है, तो इतनी बात आप यहाँ पे याद रखना. This is something you have to be very much clear with. क्या इतनी चीज आपको समझ? Are these labelings clear to all of you? Are these labeling clear to all of you?
Yes, sir.
Right? Apparently I cannot see your thumbs up right now. So please अगर हाँ yes बोलोगे ना गायत्री तो ज्यादा better रहेगा. I cannot see. So yeah, Ms. Bai is saying yes also. All right. So that is something you have to know. This is what you get to see in the dicot embryo. You have a 2 cotyledons. इनका काम होता है आपका nourishment provide करना. You have a plumule, you have a radicle. Plumule is going to be forming the shoot system in future. Radicle is going to be forming my root system in the future. So these are the few points which you have to collectively have to remember. Right? Now, अब आगे देखते हैं. Let's have a look. A typical dicot embryo consists of an embryonal axis, that is something which I have told you, and a 2 cotyledon, right? The portion of embryonal axis above the level of cotyledon, the portion of embryonal axis above the level of cotyledon is called as my epicotyle. Something I have just mentioned over here. देखो यहाँ पे. The portion of embryonal axis above the cotyledon, this area is called as my epicotyle, right? And the portion of embryonal axis below the cotyledon is called as my hypocotyle. Yeah, so ये बात यहाँ पे इन्होंने definition में लिखी हुई है, right? The cylindrical portion below the level of cotyledon is hypocotyle. So you should know what is my hypocotyle and what is my epicotyle, right? And in the lower part you have a radicle or a root tip. Okay, so there is something you have to know now. So that is how my typical dicot embryo is going to look like. So you have a plumule. You know what is my plumule is known for. कभी भी कोई पूछे plumule क्या करता है, you should know what is it is going to do, right? Then you have a radicle. Also, you should know what is my radicle is known for, and then you have your hypocotyle and you have epicotyle. You can always remember what they are going to be. Yeah, now you also have a cotyledon, which are also going to play the role in my nourishment. So that is my dicot embryo is going to look like, right? Now let's have a look at the monocot embryo. तो इसको भी हम यहाँ पे देख लेते हैं. So monocot embryo को जब आप यहाँ पे देखते हो, so you will have a single cotyledon. So here you will only find a single cotyledon. So quickly let's have a look. You have a single cotyledon in them, and the single cotyledon is called as my scutellum. This is called as my scutellum. This is called as my scutellum. Okay, so that is something you have to know. Now this single cotyledon, you can see over here called as my scutellum, is going to be seen over there, which is a single structure, single leaf-like structure, right? Now along with that there are a couple of things you have to know. You will have a plumule, तो यहाँ पे भी आपको क्या मिलेगा, you will have a plumule, you will have a radical. So अगर मैं इसका एक रफ पिक्चर यहाँ पे बना के दूं आपको यहाँ पे, so you will have again you will have over here the plumule. So the top area will be the plumule, right? This point over here is called as my plumule. Plumule चाहे monocot का हो या dicot का हो, you will find it will have a same role, right? Now this plumule will have a covering of a sheath, and that covering of a sheath is called as my coleoptile. This covering of the sheath, covering the plumule, this sheath is called as my coleoptile. Coleoptile. This sheath over here is called as my coleoptile, right? Then you have a lower part. Then you have a lower region, and the lower region over here is going to be called as your radicle. In the lower area, you will have a radicle over here. So radicle is also getting covered by a sheath. Radicle will also have a covering. So radicle के पास भी covering होती है खुद की. Protective covering radicle के पास भी होगी. And the radicle will have a protective covering, and that protective covering is called as my coleorhiza. Coleorhiza. So ये terms आपको याद रखने हैं. Coleorhiza is a covering to the radicle, and coleoptile is a covering to the plumule, and that is only seen in my monocot embryo. You can find these terms only in the monocot embryo. And याद रखना, you have a single cotyledon in them, and that cotyledon is called as my scutellum, which you can see over here. जो आपको यहाँ पे दिख रहा है. That is going to be scutellum. So इतनी बात याद रखना. You also have something like epiblast, which is rudimentary. Rudimentary का मतलब इसका कोई काम नहीं होता. Okay, तो इतनी बात आपको यहाँ पे याद रखनी है. And याद रखना, the radicle will have a root cap. Something you can understand. It will have a root cap over there. ठीक है, तो ये पिक्चर याद रखना और ये लेबलिंग याद रखना. You will find question based on this. प्लूम्यूल की कवरिंग कोलेप्टाइल, रेडिकल की कवरिंग यहाँ पे आपकी कोलोराइजा. ठीक है, तो ये चार चीजें आपको यहाँ पे याद रखनी है. नाउ लेट्स हैव अ लुक. अब जो बोला मैंने आपको ये यहाँ पे लिखा हुआ है. Embryos of monocotyledon possess only one cotyledon, right? In a grass family, the cotyledon is called as my scutellum. That is situated towards one side on the lateral side of the embryonal axis. वो आपको वहाँ पे दिख रहा था.
Sorry, your screen is not visible.
My screen is not visible. Even I cannot see
How
my screen.
visible?
अच्छा सच्ची बताओ, दिख रही है नहीं दिख रही?
How visible, sir?
है ना, दिख रही है. हाँ, right. At a lower end, the embryonal axis has a radicle and a root cap, and is enclosed by an undifferentiated sheath called as my coleorhiza. So याद रखना, you will have a coleorhiza in the lower bottom, and the upper area will have a coleoptile. The portion of the embryonal axis above the level of attachment of the scutellum is called as my epicotyle. That remains the same. यहाँ पे भी आपका epicotyle वही होगा. जहाँ पे आपका पॉइंट ऑफ अटैचमेंट है ना, जहाँ पे आपका कोटिलेडॉन लगा हुआ है स्कूटेलम, उसके ऊपर वाले एरिया को हम बोलते हैं यहाँ पे एज एपिकोटाइल, राइट? सो दिस इज अ वेरी सिंपल थिंग. इफ यू हैव अंडरस्टूड द इनिशियल पार्ट, देन दीज आर जस्ट स्टेटमेंट व्हिच यू कैन ऑलवेज रिमेंबर. ठीक है, कंफ्यूजन रखना नहीं है. सो दैट इज समथिंग यू विल गेट टू सी. Epicotyle has a shoot apex and a few leaf pyramidal enclosed in the hollow foliar structure called an epicotyle. So it's something I've told you. You will have epicotyle, and you will have outer covering of the plumule, that is my coleoptile, enclosed in a hollow foliar structure called as my coleoptile. The outer covering of the endosperm separates the embryo by a proteinaceous layer called as my aileron layer. देखो यहाँ पे इन्होंने मेंशन किया हुआ है aileron layer को. So याद रखना, aileron layer is the outer covering of the endosperm. It is the outer covering of my endosperm called as my aileron layer. So ये बात आपको यहाँ पे मालूम होनी चाहिए. ठीक है, so you can see a picture of monocot grass. यहाँ पे आपको उसका एम्ब्रियो का आपको यहाँ पे पिक्चर आपको दिख रहा है, जो हम पहले भी देख चुके हैं. यही पिक्चर आपको याद रखना है. So that is something you have to know. I think it is a very simple thing, and now let's move on to something else. तो ये हमने यहाँ पे देख लिया. Okay, now let me discuss something type like my perisperm. Okay, now what is my perisperm? Now remember one thing. You have something known as my nucellus. You know that you have something like your nucellus. Within the ovule, you have something like my nucellus. सुना है ना ये आपने, पढ़ा है ना आपने nucellus आपके ovule के अंदर होता है, right? अब जो nucellus आपका होता है ना, the nucellus you have, this is also getting consumed. This is also get consumed by the endosperm. This gets consumed by endosperm. तो एंडोस्पर्म इसको थोड़ा खा लेता है. एंडोस्पर्म इसको इसलिए खाता है ताकि वो फिर फीड कर सके एम्ब्रियो को, राइट. सो न्यूक्लियस व्हिच यू हैव इन द ओव्यूल ना गेट कंज्यूम्ड बाय द एंडोस्पर्म so that the endosperm can have a nutrient content and now that nutrient can be given to the embryo, ताकि एंडोस्पर्म के पास थोड़ा न्यूट्रिएंट आ जाए और फिर ये न्यूट्रिएंट वो दे आपके एम्ब्रियो को. तो इसलिए इसको आप यहाँ पे करते हो कंज्यूम. सो दैट इज व्हाई दिस न्यूक्लियस इज गोइंग टू इज बीइंग कंज्यूम्ड बाय माय एंडोस्पर्म. अब होता क्या है कि जनरली आपका न्यूक्लियस पूरा कंज्यूम हो जाता है. सो अगेन यू हैव अ टू कैटेगरी. You can have a category where you are getting the nucleus is getting completely consumed, or you can find a seed where you find the some amount of nucleus is been seen. If you find some nucleus is seen in the seed, if you find some nucleus is remaining in the seed, if some nucleus is seen in the seed, then such nucleus is called as my perisperm. If some nucleus is seen in the seed after the maturation of the embryo, then such nucleus is called as my perisperm. उसको हम बोलते हैं वहां पे एज अ पेरिस्पर्म. देख लो यहाँ पे. So mostly nucleus is consumed after fertilization due to absorption of the food by the endosperm, right, and the embryo. So it gets completely consumed. But in some seeds, the remnant of the nucleus are persistent in nature. कुछ seeds के nucleus का remnant आपको मिल जाएगा. The remnant of the nucleus have been seen. This residual persistent nucleus is called as my perisperm. ये जो आपको यहाँ पे residual आपको दिख जाते हैं nucleus के, you call them as a perisperm. You are going to call them as a perisperm over there. तो ये याद रखना. And now remember the example. So perisperm आपको कौन से seed में मिलेगा वो भी आप यहाँ पे याद रखो. So perisperm are been observed in my black pepper. That's one example you have to know. जिसका genus का नाम है आपके यहाँ पे piper. And the other example is my beet. I think beet भी सबने देखा है. Black pepper शायद आपने plant नहीं देखा होगा लेकिन उसको use किया आपने. So black pepper and beet are the two example of my perispermic seed. So when I tell you that it is a perispermic seed, you should know that it will have small amount of nucleus will be seen in them. Certain amount of nucleus will be clearly seen in them, and that is why we call them as a perispermic seed. So is this part clear to all of you? क्या ये वाली चीज आप चारों को यहाँ पे clear है? तनिष्क, मिसबा, शिखा and गायत्री. ठीक है? It's clear to you. I cannot see your thumbs up unfortunately for some reasons, but ठीक है. All right. Now, अब आपको यहाँ पे क्वेश्चन सॉल्व करना है. अब आपको करना है यहाँ पे क्वेश्चन सॉल्व. अब क्वेश्चन सॉल्व करने के लिए आप करोगे यहाँ पे हैंड रेज. सो पहले हैंड रेज करो. लेट मी सी इफ यू आर एबल टू सी योर रेज्ड हैंड. क्वेश्चन नंबर 7 का आंसर यहाँ पे क्या होगा? सो आई कैन सी. अच्छा देखने दो मुझे. आई थिंक हाँ चलो मुझे हैंड रेज यहाँ पे दिख रहा है. ऑलराइट शिखा, क्वेश्चन नंबर 7 का यहाँ पे क्या आंसर होगा?
ऑप्शन डी.
ऑप्शन डी होगा यहाँ पे आपका आंसर एंड दैट्स गोइंग टू बी करेक्ट आंसर. द रोल ऑफ डबल फर्टिलाइजेशन इन एंजियोस्पर्म इज टू प्रोड्यूस बोथ एंडोस्पर्म एंड जायगोट. बिल्कुल सही. ठीक है. देन क्वेश्चन नंबर 8, द फर्टिलाइजेशन अकर्स इन. हैंड रेज करो जल्दी इसका आंसर देने के लिए सब लोग. मिसबा क्या होगा इसका आंसर? क्वेश्चन नंबर 8 का आंसर क्या होगा? राइट, ऑप्शन डी बिकम्स द करेक्ट आंसर. दिस इज वेयर द फर्टिलाइजेशन ऑफ द इज गोइंग टू टेक्स प्लेस. सिंपल क्वेश्चन है. ये वाला करके दिखाओ. इन अ फ्लावरिंग प्लांट पोलुम ट्यूब फर्स्ट अराइव्स इन. हैंड रेज करो इसके लिए. सब लोगों को हैंड रेज करिए यहाँ पे. इन फ्लावरिंग प्लांट द पोलुम ट्यूब अराइव्स इन. बताइए गायत्री क्या होगा इसका आंसर?
सर ऑप्शन डी.
अरे 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 गायत्री, यू आर सेइंग ऑप्शन डी यहाँ पे. गायत्री.
यस सर.
ओके. शिखा क्या होगा इसका आंसर?
सर ऑप्शन सी.
ऑप्शन सी याद रखना बेटा. पोलम ट्यूब सबसे पहले आके कांटेक्ट में आता है आपके सिनेगेट सेल के एंड देन द सिनेगेट सेल आर गोइंग टू डाई ओवर देयर, राइट. सो सेंट्रल सेल आप नहीं बोलोगे. द पोलम ट्यूब अराइव्स इन द सिनेगेट सेल के अंदर. सो दैट इज ऑप्शन सी बिकम्स माय करेक्ट आंसर, राइट. अब ये वाला क्वेश्चन आप लोग खुद से करोगे. डू दिस क्वेश्चन बाय योरसेल्फ. इफ द नंबर ऑफ क्रोमोसोम इन द एक्स सेल इज 8, if the number of chromosome in the X cell is 8, then what is the number of chromosome in the endosperm? क्वेश्चन को ध्यान से सीख लो, समझ लो एंड देन टेल मी द आंसर. नंबर ऑफ क्रोमोसोम एग के अंदर इन्होंने बोला है 8 है. बताइए मिसबा.
ऑप्शन ए.
मिसबा इज सेइंग ऑप्शन ए. ठीक है. बाकी लोग शिखा कर रहे हो? शिखा, गायत्री.
यस सर.
बताइए क्या होगा फिर शिखा इसका आंसर। एंड अगेन प्लीज रेज योर हैंड बिकॉज आई कैन नॉट सी यू अगेन। कोई तो भी हैंड रेज करना एक बार फिर, प्लीज एनीवन रेज योर हैंड। अरे जस्ट रेज योर हैंड दैट, हाँ या, थैंक यू मिसबा। हाँ जी बेटा बताइए गायत्री शिखा क्या होगा इसका आंसर। इतना टाइम कैसे लग रहा है बेटा, इतना टाइम इतना। चलो ले लो, ठीक है अब आपको
सर
एक्सप्लेन करना पड़ेगा। बोलो बोलो।
ऑप्शन ए।
ऑप्शन है कैसा है, ऑप्शन है बताना।
सर एग सेल जो है 8 है और जो स्ट्रक्चर बनेगा एंडोस्पर्म का वो 3N होगा,
हाँ
तो फिर 24 प्रो में।
राइट, सही बात है। आपका एग यहाँ पे है हैप्लॉइड एग दैट्स गोइंग टू बी हैविंग द 8 क्रोमोसोम्स। और आप जानते हो कि आपका एंडोसोम आपका होता है हमेशा आपका 3N होता है। सो 8 + 8 + 8 एंड दैट मेक्स इट माय 24 क्रोमोसोम्स। गायत्री पता चला एंड ऑल द अदर पीपल, ठीक है। ऑलराइट नेक्स्ट, ध्यान से देखो इस क्वेश्चन को। डबल फर्टिलाइजेशन इन्वॉल्व्स फर्टिलाइजेशन ऑफ, डबल फर्टिलाइजेशन इन्वॉल्व्स फर्टिलाइजेशन ऑफ, बताओ बताओ इसका आंसर बताओ। हैंड रेज करना पहले एंड आंसर बताइए। गायत्री क्या होगा इसका आंसर। और जिसने हैंड रेज किया टेल मी द आंसर।
ऑप्शन डी।
सर ऑप्शन
ऑप्शन डी, ऑलराइट। अगेन प्लीज ट्राई टू रेज योर हैंड अगेन, जस्ट आई कैन नॉट। ओके या, हाँ जी मिसबा बताइए क्या होगा इसका आंसर।
ऑप्शन डी।
ऑप्शन डी, यस ऑप्शन डी बिकम्स द करेक्ट आंसर। देखो यहाँ पे क्या हो रहा है, डबल फर्टिलाइजेशन मींस व्हाट, the egg is involved, so you have an egg, you have a central cell and they are getting fused with what, two sperms. So दो sperm आ रहे एक egg के साथ fuse करा, एक central cell के pollen nuclei के साथ fuse करा and brought by the same pollen tube. So that is why option D becomes my correct answer. ठीक है, अब next question करेंगे हम थोड़ा सा ध्यान से। अब यहाँ से आपके नीट के क्वेश्चन स्टार्ट। नाउ यू कैन सी दिस, these are my NEET questions, so now please try to solve them. Okay, what is the fate of a male gamete discharged in the synergid? What will happen, what is the fate of a male gamete when they have been discharged in the synergid cell? Now take your time, take your 30 seconds and then raise your hand. All right, Gayatri, tell us.
सर ऑप्शन ए।
What do you think, Misbah
ऑप्शन ए।
and Shikha?
सर ऑप्शन ए।
Yes, option A becomes my correct answer. Option A over here will become my correct answer. Okay, right, very good. Double fertilization is what? Read the statement and tell me the correct answer. देख लो, ये 2018 का क्वेश्चन है। क्या होता है डबल फर्टिलाइजेशन के दौरान। टेल अस गायत्री।
सर ऑप्शन डी।
ऑप्शन डी, ओके बाकी लोग। मिसबा
ऑप्शन बी।
एंड शिखा।
सर ऑप्शन डी।
राइट, सिंगैमेट रिवोल्यूशन, those two things will happen over here, right? This is the correct answer. बाकी कोई भी आपका यहाँ पे सही नहीं है। यहाँ पे क्या लिखा है, 2 male gamete with egg, ऐसा नहीं होता। Fusion of 1 male gamete with the 2 polar nuclei, that doesn't happen. So that is why option D is the best answer over here, right? बाकी सब आपके यहाँ पे गलत। क्वेश्चन नंबर 84, अनम्यूट करके जोर से आंसर वैसे ही बोल दो, अनम्यूट करके जोर से।
एंजियोस्पर्म।
Right. Angiosperm is going to be a correct answer. That is something specifically seen in my angiosperm. That's my double fertilization. अब एक बात यहाँ पे याद रखना, एंजियोस्पर्म के अंदर आपका एंडोस्पर्म जो होता है, सो याद रखना एंडोस्पर्म जो आपका होता है एंजियोस्पर्म के अंदर वो कैसा होता है, कैसा होता है। अरे डिप्लॉयड होता है, टेट्राप्लॉयड होता है, कैसा होता है एंडोस्पर्म।
टेट्रा।
अरे डांट खाओगे आप लोग। एंडोस्पर्म कैसा होता है बाबा। हाउ इज एंडोस्पर्म? डिप्लॉयड होगा, हैप्लॉयड होगा, कैसा होगा एंडोस्पर्म? एंजियोस्पर्म के अंदर ट्रिप्लॉयड होगा ना, ट्रिप्लॉयड होगा। ऐसी चीजें भूलोगे तो कैसे चलेगा, राइट? ये बात याद रखनी है आपको यहाँ पे। जिम्नोस्पर्म में क्या याद रखना है आपको? जिम्नोस्पर्म के अंदर जो आपका एंडोस्पर्म होता है, द एंडोस्पर्म यू विल फाइंड इन द जिम्नोस्पर्म। रुक जाओ। The endosperm you will find in the gymnosperm. This endosperm is going to be always going to be haploid. ये बात लिख लो, भूलना मत, ये क्वेश्चन आ सकता है। जिम्नोस्पर्म के अंदर भी एंडोस्पर्म बनता है, बट दैट इज बीन ऑलवेज गोइंग टू बी माय हैप्लॉइड इन नेचर। ओके, क्या इतनी बात आपको याद रहेगी? क्या इतनी बात आप समझ पा रहे हो, आप चारों तीनों लोग यहाँ पे?
Yes, sir.
ठीक है। हाँ, अब एक बार इसमें और ऐड करो, ऐड वन मोर थिंग इन दिस। वन मोर थिंग व्हाट यू हैव टू नो हियर इज that endosperm in gymnosperm is formed before fertilization. So the endosperm is getting formed over here before fertilization. So even before the sperm comes, even before the male gamete is going to come, before that the endosperm is getting formed. So before fertilization, the endosperm is getting formed, right? Here the story is different. Here in angiosperm, what has been happening? It is happening only after fertilization. It is happening after fertilization. Okay, that is something you have to know. Which of the following statement is wrong? So now read the statement carefully and tell me which statement over here is going to be incorrect. Question number 85. Tell us, Shikha, कौन सा incorrect है?
सर ऑप्शन ए।
मिसबा, आपको क्या लग रहा है?
Option A.
Right, and I'm very sure गायत्री को भी लग रहा होगा यही. When pollen are shed at a two-cell stage, double fertilization does not take place. You don't find double fertilization taking place over there. ये गलत बात, ऐसा नहीं होता, ऐसा नहीं होता आपका, वो तो होगा ही होगा, डबल फर्टिलाइजेशन आपका होगा, बिकॉज टू सेल स्टेज से थ्री सेल स्टेज आपका बन जाता है. You just need one mitosis and you'll find two male gametes have been formed. So that is why option A is going to be a incorrect statement. 2012 का ये था आपका यहाँ पे क्वेश्चन. Now, through which cell of the embryo sac does the pollen tube enters the embryo sac? अगेन सिंपल सवाल है, सब लोग अपना हैंड रेज करेंगे यहाँ पे. कौन से सेल के थ्रू इन द एम्ब्रियो सैक डज द पोलन ट्यूब एंटर्स? गायत्री वेटिंग फॉर यू. टेल अस, क्या होगा इसका आंसर? अच्छा, एक बात याद रखना गायत्री, हमेशा आपका पोलन ट्यूब एंटर करता है किसी चीज को ब्रेक करके. सो दरवाजे में कौन खड़ा होता है? माइक्रोपाइल के दरवाजे में, इन द एट द डोर स्टेप्स ऑफ द माइक्रोपाइल यू हैव सम सेल्स स्टैंडिंग ओवर देयर. व्हिच आर दोस सेल, गायत्री, व्हिच आर बीन स्टैंडिंग नियर टू द माइक्रोपाइल? I think Gayatri is...
Sir, can I repeat, sir?
Okay, see, what will happen now, you know that at a macropyle region, you will find there are some cells standing over there. Those cells are being broken down by the pollen tube, and those cells are my synergate cells, right? So when they ask you through which cell of the embryo sac does the pollen tube enters the embryo sac, then the answer is going to be my synergate cells. So degenerated synergate cells को हम वहाँ पे खत्म करेंगे, and then you are going to enter inside the embryo sac. So option C over here becomes my correct answer, right? मिसबा, शिखा, आपने भी यही चूज करना था, है ना?
Yes.
All right, चलो ठीक है. Then अब देखो ध्यान से क्वेश्चन, सब लोग ध्यान से देखो इस क्वेश्चन को. Generative cell was destroyed by a laser. Generative cell was destroyed by a laser, but a normal pollen tube was still formed because generative cell was destroyed by a laser, but still the normal pollen tube was formed. Tell me how that happened. Read the statement carefully and try to answer. It's a very simple question actually. ऐसा घुमा के पूछा, but है simple. Generative cell was destroyed by a laser, but a normal pollen tube was still formed because. You have A, B, C, D. Now tell me which can be the potential answer over here. Tell us, Misbah.
Option A.
मिसबा सिंह ऑप्शन ए। शिखा आपको क्या लग रहा है?
सर ऑप्शन डी।
Okay. Now let me tell you. The region of emergence of pollen tube is not harm. That is not a very appropriate answer. I'll tell you why it's not appropriate answer. Because you have an option A over here which says that vegetative cell is not damaged. In the question, what they have said that which of this cell, like they have said the generative cell is born destroyed by the laser. तो अगर generative cell गया, so कोई बात नहीं, pollen tube तो बनेगा ही बनेगा. Pollen tube formation is happening from the vegetative cell. So you don't need a presence of a generative cell for that, right? So that is why option A is the most appropriate correct answer. Not option D, not option C, not your option B, right? ये आपका जिंदा था. The vegetative cell was alive over there, and because of that, you were finding that pollen tube was successfully formed. पता चला? शिखा समझ आया?
यस सर.
ठीक है, तो दैट वाज माय ऑप्शन ए एंड द क्वेश्चन गोस टू गायत्री. गायत्री, क्लियर है ये क्वेश्चन आपको? इज दिस क्वेश्चन क्लियर टू यू?
Yes, sir.
गायत्री, व्हाई इट सीम्स लाइक यू आर इन सम काइंड ऑफ वर्क राइट नाउ? भागम दौड़ी कर रहे हो, कुछ क्या हो रहा है गायत्री? व्हाट इज हैपनिंग? नथिंग.
Sir, can you repeat? Sir, I don't understand.
I am asking, are you running somewhere? Are you occupied in somewhere? You are not able to engage in the class. That's what I'm asking. What is happening? Are some guests in your house? What is happening?
No, sir.
All right, okay, ठीक है. Now this is a very good question you have to think and solve. Total number of meiotic division required for forming 100 zygotes. Total number of meiotic division required for forming 100 zygotes or 100 grains, whatever, it is the same thing, of wheat. Now you think and tell me how many meiotic division I need to have. ये करना पड़ेगा, आराम से इसे करना पड़ेगा. You do this question and tell me. मिसबा ने कर दिया बहुत जल्दी. बताओ मिसबा, क्या होगा इसका आंसर?
Option D.
Option D, Misbah. Misbah, please cross check again. Is your answer completely true or not? बाकी लोग भी करो. Take your 30 seconds, take your 1 minute, 2 minute, and try to solve this. Question is very clear. You have to make 100 zygote. You tell me how many meiotic division you need to have. Misbah, do again. Huh? You have to do it again. वापस एंड्रेस किया. बताओ.
Option A.
Okay. बाकी लोग भी ट्राई करिए. Shikha and Gayatri also, you also try. Because of a network issue, Tanishq is not there in the call. शिखा, गायत्री, हिंट चाहिए या कर रहे हो? कर रहे हैं. Misbah, you have to do it again. Misbah, do it like आराम से करो उसे, so you'll get the answer. Gayatri, what is happening? Trying?
Yes, sir.
Okay. Okay, now it's like too much time now. It's too much time. Tell us something. शिखा बताओ क्या आंसर आ रहा है?
So 50.
Can't hear you, शिखा. Can't hear you. जोर से बोलो.
सर ऑप्शन ई आ रहा है.
ऑप्शन नहीं आ रहा है. हाँ, अब देखो आवाज आई आपकी. गायत्री, इस बार आपका ऑप्शन नहीं आ रहा था. गायत्री, आपका क्या आ रहा है? What is your answer, गायत्री? Say something.
So option B.
Option B. All right. Okay, now I'll solve. And to solve this, I need some space. Where is the space? This is the space I will use. Okay, now. So I have to form 100 zygotes. I have to form 100 zygotes. Now, if I have to form 100 zygotes, there is one thing should be very clear to you, that I need to have 100 sperms, right? I need to have 100 sperms. I need to have, or I will make it slightly more simple to you. If I want to make 100 zygotes, I need to have two things. I need to have 100 pollen grains, and I also need to have 100 eggs. Is this clear to all of you? शिखा एंड सबको, तीनों के इतना बात समझ आ रहा है? मुझे 100 पोलन ग्रेन चाहिए होंगे और मुझे 100 एग चाहिए होंगे. ये बात क्लियर है सबको? इसको लेके कंफ्यूजन हो तो पूछ लो. If I have to make 100 zygote, I need to have 100 pollen grains and I have to make 100 egg. Okay. Now let's come back over here. If I have to make 100 pollen grains, अब देखो यहाँ पे 100 pollen grains आप बनाते कैसे हो? You have a diploid MMC. Diploid MMC. Just wait a minute here. You have a diploid MMC over here. This diploid MMC will undergo the meiosis and I will give my 4 macrospores. I will get my 4 macrospores have been formed. Just wait a minute. Class, you just have to wait a minute. Huh? Just wait a minute. Let me plug in the charger. तब तक मैंने आपको थोड़ा हिंट दिया है ना, उसके ऊपर काम करो. Just give me a minute. I have to plug in the charger over here. All right. Okay. ठीक है. So you need to have 100 pollen grains and you need to have 100 eggs. ये आपको चाहिए होंगे so that you can make your 100 zygotes. तो अगर आप जानते हो, अगर मैं एक बार meiosis करता हूँ यहाँ पे, if I do one time meiosis, I get 4 macrospores, right? I get 4 macrospores. So basically means what? इसका मतलब यहाँ पे क्या होता है? इसका मतलब यहाँ पे ये होता है, it means what? That when I do one time meiosis, when I do one time meiosis, I get 4 pollen grains, right? So if I now want to have 100 pollen grains, तो मुझे कितने टाइम करना पड़ेगा meiosis? बताओ मुझे.
25.
25. ठीक है. So 25 टाइम्स जब मैं करूंगा अपने यहाँ पे meiosis, so I will have my 100 pollen grains have been formed. तो ये तो मुझे यहाँ पे मिल गया. तो एक आंसर मुझे यहाँ पे मिल गया. One part is clear to me that I have to carry out my meiosis 25 times. So 25 times meiosis मुझे यहाँ पे करना पड़ेगा. ये चीज तो मुझे यहाँ पे क्लियर है. इतना तो पहले मुझे करना ही पड़ेगा. Right? Now you have 100 eggs. Now tell me something. If you have to form the 100 egg, now understand this thing. You have a megaspore mother cell. Assume that you have a megaspore mother cell. Right? That's my MMC. Now this MMC, assume that will undergo something which you called as a meiosis again. It's a megaspore mother cell. It will undergo the meiosis. अब आप मुझे बताओ, जब ये meiosis करेगा, you will have 4 cells have been formed. 4 haploid megaspores have been formed. Now tell me something. Out of these 4 megaspore, how many will survive? कितने survive करेंगे यहाँ पे?
1.
One.
Right. So 1 will survive and 3 will degenerate. So 3 of them are going to degenerate and 1 of them is going to survive. Now tell me something. अगर ये मेरा यहाँ पे survive कर रहा है, so this megaspore will make the embryo sac, right? You know that it will undergo the 3 times mitosis and will give you the embryo sac. Will give you the embryo sac. Now tell me, in a single embryo sac, कितने egg होते हैं single embryo sac के अंदर? How many egg you have in a single embryo sac? बताओ. एग सेल कितना होता है एक एम्ब्रियो सैक में? कितने एग सेल होते हैं? 1. इतना क्यों सोचते हो आप लोग? इतना नहीं सोचना. एक सिंगल एम्ब्रियो सैक में यू विल हैव a 1 egg cell. You will have 1 egg cell, right? Now think in this way. When you do one time meiosis, you get 1 egg cell. When you do one time meiosis, you get 1 egg cell. So if I want to have 100 zygote, I need to have 100 eggs. तो अगर मुझे 100 egg चाहिए होगा तो मुझे कितने बार meiosis करना पड़ेगा?
100 times.
100 times, right? तो यहाँ पे मुझे 100 टाइम करना पड़ेगा meiosis और यहाँ पे करना पड़ेगा मुझे 25 times. तो टोटल मेरा कितना हो जाएगा? 125 times. Total will be over here my 125 times. समझ आया कैसे आया 125 यहाँ पे आंसर?
Yes, sir.
जब तक खुद से करोगे नहीं, until you do it by yourself, until Gayatri, Shikha and Misbah does not do this question by themselves, until then you won't understand. So you have to solve this question by yourself in the notebook, right? After lecture, after having your dinner, please make this question by yourself. Have that calculation again by your own hands. Then only you will remember this question. Okay? That is something you will find happening over here. Okay. Now tell me this answer for this question. Take your time. Take your 20 seconds, 30 seconds, then raise your hand. Endosperm is formed during the double fertilization by. All of you should raise your hand. Endosperm is formed during double fertilization by. शिखा क्या हुआ? मिसबा बताओ क्या होगा इसका आंसर? शिखा एंड गायत्री.
So your screen is not visible to me.
स्क्रीन नहीं दिख रही मेरी? अरे अरे अरे, अभी भी नहीं दिख रही? शिखा अभी भी स्क्रीन नहीं दिख रही?
No, sir.
अरे व्हाट हैपेंड? गायत्री आपको स्क्रीन दिख रही है?
Yes, sir.
Okay. Shikha, why you cannot see the screen? You should be able to see the screen. आपको अभी भी कुछ नहीं दिख रहा?
No,
शिखा
sir.
नहीं दिख रहा अभी भी? All right, that's kind of strange. Just wait. Yeah, Gayatri, tell me क्या होगा इसका आंसर?
So option A.
I'm going to give you the correct answer in this. Now let me just come back over here, Shikha, and let me again start the screen share. Just wait a minute. All right, रुको, वापस हम स्क्रीन शेयर करते हैं यहाँ पे. शिखा आप बताइए, अब स्क्रीन दिख रही है आपको?
No, sir.
अभी भी नहीं दिख रही? Then there should be some network issue on your end, Shikha. Possibly network issue on your end. क्या दिख रहा है? सब कुछ ब्लैक दिख रहा है?
Yes, sir. दिख ही नहीं रहा है.
अच्छा, तो there must be some network issue on your end. ठीक है. Alright, कोई बात नहीं. So let's, वैसे भी आपने यहाँ पे almost time यहाँ पे हो चुका है. So let's keep it up to here. I would strongly recommend कि इसके अंदर और भी यहाँ पे कुछ क्वेश्चन लगा के रखे हुए हमने यहाँ पे. Please solve the remaining 3 questions from your end and फिर मैं आपके साथ assignment share करूंगा. ठीक है. अब ये तो चलो ठीक है, 2 मिनट रुक जाओ before you leave the class. So there is something we have done. So in today's class, remember we have done the 2 type of embryo. We have done the monocot, we have done the dicot, we have seen the embryo development or embryo, what are the stages of embryo development, and then we have understood everything in terms of the endosperm. ये सब चीजें हमने यहाँ पे देख ली along with my albuminous and ex-albuminous seeds. That is something we have all seen over here. Now just wait a minute. अच्छा, एक सेकंड रुक जाओ. All right.